Hi, Assalamualaikum. I am Reina Sarah, and in this video I will be discussing about DNA sequencing. So DNA sequencing is a determination of the order of the four chemical building blocks called bases that make up the DNA molecule. This is according to the NIH website from the 2020. So why do we do sequencing? It is to determine the order of amino acids in nucleic acids and to further explore the function of sequenced genes and understand the genetic information carried besides determining the complete nucleotide sequence for each chromosome. So using Sanger sequencing method, we first anneal the DNA using heat to separate the double-stranded DNA into a single-stranded DNA as the template strand for the DNA sequence. Next, the single-stranded DNA will be annealed and attached to a primer at the end of the single strand. After that, the annealed strand will be distributed into 4 different polymerase solutions that includes DNTPs, DNA polymerase, and small concentration of DDNTPs. So DNTP and DDNTP are building blocks of DNA, except DDNTP lacks 3-OH, which terminates the transcription by DNA polymerase. So DNA chain will be terminated without the 3-OH. DDNTP will stop the DNA polymerase reaction. After that, the DNA polymerase will bind to the DNTP to the template strand until it reaches the DNTP and stop. The termination resulted in DNA fragments in different lengths. The 4 solutions are then brought for gel electrophoresis, and the DNA will be moving towards the positive pole. It will also follow their according size, the lighter, the smaller, the further. We then read the DNA sequence according to the pattern from the bottom. In this diagram, we read it as C, A, G, C, G, A, T, T, A, C, G, C, T, and so on. That is all for me. Thank you.
Created using Powtoon.